Alright guys, so we're on meiosis, which is basically just part 2 of cell division. Meiosis is a little different from mitosis. I'll give you the whole list at the very end. But basically we're going to talk about the formation of gamete or sex cells. Mitosis deals with somatic cells slash body cells. We're going to talk about how to basically make the egg and the sperm. So most adult organisms contain 2 sets of inherited chromosomes and 2 sets of completed genes. The chromosome, again, we went over to meiosis. You're going to basically have strands of DNA and protein inside the nucleus, and it's going to carry the genes. When you're looking at the chromosomes, they're going to be wrapped up with DNA, basically. The genes are going to be located in specific locations. So if I'm looking at a chromosome for a human, maybe this is where hair color is. It may not be the same for another organism, but each gene basically has a designated spot where it's going to be located. So meiosis, like I said, is basically a formation of sex cells. Now, if you have your full set of genes in an egg, you wouldn't need to be fertilized by the sperm. So a diploid cell is not going to cut it for meiosis, because again, we are trying to form those sex cells, because you're going to get half of your information from mom and half of your information from dad, which means we need half of the information in each in order to make you. So instead of having a diploid cell, we're going to end up with 4 haploid cells. Now, I can't give you a universal number of diploid or haploid cells because it varies from organism to organism. So when we're talking about diploid cells, we're going to mark it as 2N. Oops, I know it's not cute, guys, but D2. And for meiosis, like I said, we're going to have haploid cells. Haploid, think half. Half of 2N is just N. So this is our genetic formula for diploid and haploid cells. In order to get that haploid cell that we need, though, meiosis is going to need to go through cell division twice. So we went over it before. Mitosis has basically PMAT, and meiosis is just going to have PMAT twice. Very important to note, interphase is going to happen here. It does not happen here. There is no interphase because we're not trying to go back to the diploid cells. We want that information to be cut in half. So you're going to have PMAT 1 and PMAT 2. Again, no interphase in the middle. Alright, so again, interphase, basically the cell getting ready for cell division. You're going to have it replicating into DNA, ready to go into 2 different cells. Then you're going to go to prophase, where the DNA is going to condense and you're going to create that tetrad, which again is just the X that we're going to model for you guys. Each contains 4 chromatids, 1, 2, 3, 4. Now the nuclear envelope is going to dissolve, so you're going to have your free-floating X's. Now, what makes meiosis so different from mitosis is, again, this is meant for reproduction. So we want to basically make sure we get as much genetic diversity as we can, and this step is one of the most important ones to get that. What happens basically is that your cells will cross over, again why it's called crossing over. They'll basically touch little legs and then they'll separate their caps. So they'll switch socks basically. You'll see it here, the chromosomes are different. They need to be homologous though, which means they need to code for the same thing, which means if I changed, let's say eye color, that's not how that works, but let's say eye color. Here, this one must also code for eye color, so we're switching the eye color gene. We are not creating a new one, we're not putting hair color where eye color is going to be. That's why they need to be homologous, which again just means the same. So once they go through crossing over, we're going to go through the usual steps. Metaphase, M for middle, they'll meet in the middle. Anaphase, A for away, so they'll be split apart. It's the same as mitosis. So then you're going to go to telophase I, which the nuclear membrane is going to form again. You're going to have your clusters of chromosomes. Cytokinesis comes where it just pinches in the middle, getting ready for that cell division. In plants, it's going to create that cell plate instead of a pinch because, again, they're rigid and they have cell walls. And then you're going to have your 2 daughter cells. So these are your diploid cells. Again, we do not want diploids, which means we have to do it one more time. So we're going to enter PMAT 2. Prophase, crossing over does not happen in Prophase II. Please make sure you understand the difference. If I'm showing you crossing over and asking you what stage this is at, do not tell me Prophase I and II because that is wrong. crossing over only happens once, and that is in prophase I. This is going to be the same as mitosis. You're just going to have the nuclear membrane leave, the tetrads form, and then we slowly start the cell division. Metaphase, M for middle, Anaphase A for away. So instead of having your X's in each cell, now you're going to have half of the information. And that's how you're going to end up with your 4 cells. We're not at the full cell division yet. I just want you guys to see what it's going to look like at the end so you don't get confused. Telophase, again, they start to pinch. Cytokinesis, they finally get severed. So this is how we're going to go for the creation of sperm. For guys, because life is always so hard, you can make sperm for forever. So you make 4 cells, these will all become sperm. Girls, it's going to be different. So you'll have 4 cells, however, because we like to be complicated, one of these will become a viable egg, the other 3 will basically become cell food. It's going to become junk. Only one of them is actually viable, if not you would have unlimited amounts of eggs, and we have a limited amount of eggs from birth. Again, okay, the differences between meiosis and mitosis. Meiosis is meant for genetic diversity and any form of sexual reproduction. You're going to end up with different daughter cells. Mitosis, you're going to end up with almost clones of each other, and it's made more for growth and repair than anything. So as you grow, you go through mitosis. You cut yourself, mitosis will make sure that you have a new layer of skin later on. Mitosis does not change the number of chromosomes in the original cell. Again, it basically just makes a carbon copy of itself. Mitosis makes a copy of itself, and you end up with 2. Meiosis, you end up with 4. They're all genetically different, and they are haploid cells. Mitosis can be used as a form of asexual reproduction because, again, you're just cloning yourself. Meiosis is made for the first steps of sexual reproduction. Again, it is not sexual reproduction until the gametes have combined. And I'm pretty sure this is just going to go over exactly what I said, so our lecture is over.